Welcome to our webinar titled Genetics and MDS. Thank you for joining us. My name is Lee Clark, Director of Patient Services, and I'll be moderating the presentation today. As we get started, I'd like to recognize the generous support of our corporate sponsors, Amgen, Alexion, Apellis, Taiho Oncology, and Merck. And I'd also like to recognize the generous support of our patients, families, and caregivers for supporting the program today. Before we start with the webinar, I'd like to let you know about a few educational opportunities that may be of interest to you. We have 3 patient and family conferences left this year. Our upcoming conference is with the 3 Mayo clinics located in Phoenix, Arizona, Jacksonville, Florida, and Rochester, Minnesota. That conference will be held on Saturday, September the 23rd, and you can register directly on our website at aamds.org. Also, we're hosting a patient and family conference, which will be on Saturday, October the 14th, in New Haven, Connecticut, and that conference is in partnership with Yale Cancer Center. Our last conference of the year is in Dallas, Texas on November the 4th. That conference is in partnership with UT Southwestern Medical Center. All of the conferences you can register directly on our website at aamds.org. Next, I'd like to let you know about our last March for Marrow walk, and that walk is being held in Virginia on October the 1st. If you can't join us in person for the walk, you can also register and join us virtually. We appreciate everyone's support in raising awareness for bone marrow failure diseases with our walks. I'd like to go over a little bit about how you'll be asking questions for the presentation. If you look to the right on your engagement panel, you'll see a Q&A icon. All you need to do is click on the icon, and then you're able to type in your question or comment. Once you have finished typing, just hit enter or the submit button, and your questions will come to us, and we'll be filtering through those questions and asking the questions at the end of the presentation. When asking questions, I do ask that you do 2 things to help us manage all of the incoming questions. First, submit all of your question at the same time. You can submit multiple questions, but make sure when you submit each question, the question is full, so we don't miss any of the important information that you'd like to present to the presenter to be able to answer your question in the best way possible. Second, please do not share any private health information in your question. Our speaker cannot answer any specific questions related to your personal health care. I'd also like to let you know that our speaker today is a board-certified genetics counselor and not a medical physician, so she will not be able to answer any specific questions related to your diagnosis or treatments. So I just want to make sure everybody understands that. So today, it's my pleasure to welcome Jennie Vagher, who is a board-certified genetic counselor who graduated from the University of Utah's genetic program in genetic counseling. She joined the Family Cancer Assessment Clinic at the Huntsman Cancer Institute in 2017. She led the development of the pediatric bone marrow failure program and the adult bone marrow failure program for patients with inherited bone marrow failure syndromes and other predispositions to hematologic malignancies. She provides genetic counseling services to patients across Utah, including those seen at Primary Children's Hospital and at the LDS Hospital. She is an expert in rare cancer predisposition syndromes. It's my pleasure to welcome Jennie today.
Thank you so much, Lee. I'm going to go ahead and share my screen with everybody. All right. Well, I consider this a success that we even got my screen shared appropriately, that it's a Friday morning. So I am just so happy to be here with everybody talking about one of my favorite topics, which is the genetics of blood cancers. So thank you so much to the Aplastic Anemia MDS Foundation for having me here today. And I just look forward to going through this information with everybody. So as Lee mentioned, my name is Jennie Vagher. I'm a genetic counselor at Huntsman Cancer Institute. This is absolutely one of my favorite photos of the Valley and of the University of Utah. And Huntsman is, tucked right in here to the hill. I joke with patients frequently when I'm seeing them when they're admitted that, we've got great mountain views because we're on the side of a mountain. So the Valley looks a lot like this right now going into the fall. So anyways, so just to get started, I thought I would start with one of my favorite phrases that I share with patients, which is that all cancer is genetic, but not all cancer is hereditary. So what we mean by that is genetics really make up how cancers develop. And, we often talk about when we're able to look at a cancer under a microscope or whenever we're able to do genetic testing for a cancer diagnosis, it's really just an accumulation of genetic mutations or errors within our DNA. But, just because those accumulation of errors exist, that does not mean that you were born with them or that those are something that you can pass down. So that's really where we're making this designation that not all cancer is hereditary and only a portion of cancer is hereditary. So I hope that by the end of this presentation, you'll have a better scope of how MDS is genetic and the situations in which it can be hereditary or be passed down through families. I often like to just give everybody a primer. I never know, how much genetics people know. And so I really just like some of these images. So I'm going to start by, giving you a primer to genetics and then we'll get into the more advanced learning here. So this beautiful picture here is a representation of the double-stranded DNA helix. So our DNA is made up of base pairs that are, the base pairs that are building blocks to life. So we often talk about base pairs being A's, T's, G's, and C's. And so all of these come together to eventually make our proteins. And then, our proteins are what actually make our body function like it should. And so then, what we also know is that as DNA replicates, so, we start out, especially in our bone marrow or, even as little tiny embryos, we start out as one cell and then that one cell divides. And so this picture here is really just representing what DNA replication looks like. And so when you start out with DNA replication, in this little cell up here, there's a whole process that happens in order for one cell to turn into two cells. And the machinery that replicates DNA is so advanced. Sometimes it really blows my mind. And I do this every day for a living that, that we're able to replicate our DNA without any sort of mistakes happening. But we do know that every time a cell divides, which happens thousands and thousands of times in our body, there is an opportunity for a mistake or a mutation, or we often call them variants, to occur. And often those mistakes occur on a single level where, maybe one base pair instead of a base pair gets switched to a G, or sometimes it happens on a much bigger level where maybe there's some slippage that occurs during replication. And now all of a sudden, you have a whole entire piece of your chromosome missing. And like I mentioned, this happens thousands and thousands of times in the body. And, we know it happens thousands and thousands of times in the bone marrow because we're constantly trying to make new, red blood cells and platelets and white blood cells. And And so, our body though has the ability to fix these DNA mistakes or these mutations. So this picture here is really just showing, that there's lots of different ways that we can develop DNA mistakes. So sometimes those mistakes are just what we call endogenous. So these are mistakes that just occur by the process of, replicating DNA. And sometimes these are exogenous. So, where we have things like radiation or chemo, or, we know that skin cancers are caused by, the accumulation of sun exposure throughout our lifetime. So there are lots of different ways that, we accumulate DNA mistakes, but the beautiful thing is that we are usually able to repair that. So there's multiple different pathways in which this repair can take place. But, sometimes we're not able to repair that. And sometimes what ends up happening is that we then accumulate genetic mutations. And so, let's say the first mistake happens because our body doesn't catch the error that happens as our cells are dividing. And then, those cells continue to grow and proliferate, and then we develop a second mutation. And maybe that second mutation is because of acquired damage because we've had a previous cancer, or maybe it is we work in an occupational exposure, or maybe it's just because we live on this planet. And then, we develop third mutation, and then, all of a sudden it's easier for our body to escape that machinery that's supposed to fix it. So maybe this third mutation is turning off a gene that is supposed to be doing this double checking in our body. And so now all of a sudden we just have this sort of beautiful random occurrence of events that then develops into cancer. And that is really the basis of everything that we know about MDS and everything that we know about how cancer develops, is that it's just an accumulation of genetic mutations or genetic mistakes that happen over time. And then this picture right here is representing this on a cellular level in the bone marrow. So what you have up here is you have your stem cells, and your stem cells make your progenitor cells, which are the precursors to our red blood cells and our platelets and our white blood cells. And so what happens then is that these progenitor cells are made in our bone marrow, and then they're pushed out into our peripheral blood, so our circulatory system, so that our body can then, use the platelets to clot, or use the red blood cells to transport oxygen to the rest of our body from our lungs, or fight infections. And so what happens though in MDS is that, these genetic mutations happen within the bone marrow, and often happen within these stem cells. And so then now these blasts, I've heard somebody describe blasts as just these like immature little kindergartners who can't really perform much function except for they just kind of take up some space in the bone marrow, take up some resources, and they're not able to mature and become productive members of the society, or the society in our body. So these blasts then kind of hang out in the bone marrow here, and they're just accumulating genetic mutations. And then instead of being pushed out into the rest of the body to make, our platelets and, to perform these functions that we need, they're not able to do that. And so then that's when we see that decrease in red blood cells and platelets and white blood cells. And so, what we know is that accumulated genetic mutations in the bone marrow define MDS. So when we look at the patients that have developed MDS, 90% of patients actually have at least one mutation that defines their MDS. And so this graph here is just representing, the most common genetic mutations that define MDS, like TET2 or SF3B1. And I will just mention here that this picture is showing the different types of MDS, and it is a little outdated. As the terminology of different types of MDS is constantly changing. And so this one is a little bit older, but the bottom line is that, many different genetic mutations are what defines MDS. Now, how do these genetic mutations develop? So, is it something that we did or didn't do in our lifetime? That is always the question. And so I'm going to go through a couple of these different opportunities for genetic mutations to develop over our lifetime. So the first is age. So MDS is associated with older age. We know that, as I've showed that, it doesn't take just one genetic mutation. It doesn't take just two genetic mutations. It is multiple genetic mutations, and that often takes place over the course of somebody's lifetime. And so, that is really a defining feature of MDS, is that, we see it at an older age because it takes a while for those genetic mutations to develop. So the median age of diagnosis is about 72 years old. Now, I will just also mention that this is not well defined here on this slide, but MDS is considered a rare cancer. So we talk about other cancers like breast cancer or prostate cancer, and we know that those are much more commonly diagnosed cancers. So when we see MDS, we don't see it often, but if we do, we see it in individuals who are typically over the age of 70. And so we do see it at younger ages. So we do see young adults with MDS, and we do see pediatric patients with MDS. But as you can see here, those are much, much more unlikely to develop during the ages under the age of 50. Now, we know that MDS may also be associated with previous environmental exposure. So benzene exposure is often one that gets talked about, where we initially, noticed and we observed that in Turkish shoe workers in the 70s, that some of these patients developed bone marrow failure, with some having this sort of pre-leukemic state, which is how we often used to describe MDS. We now have exposure limits for benzene in the modern Western industries. So, working in shoe factories or other factories where benzene is a common solvent that's used. But, we do still recognize that this could be an exposure that leads to MDS. We also know that tobacco use is associated with an increased risk for MDS, and we think that it's most commonly associated with having, current smoking habits, but we think that the risk could be increased for, up to 10 years, maybe 15 years after you quit smoking. And the good news for those that have quit smoking is that, eventually, like I said, 10 to 15 years after you quit smoking, your risk eventually does return to baseline risk. So our body is able to sort of compensate with that. But I always tell people, try to avoid smoking, because yes, it can increase your risk for MDS, but it can also increase your risk for lots of other cancers. And then there are other exposures, like, there have been documentation of exposures to, hair dyes and other chemicals that can potentially increase the risk for MDS. But exposure history is very hard to study, because unless, we get some sort of single event where we can study it by itself as a single variable, like, Chernobyl, for example, then it becomes really hard to, control for other variables. Who's to say that the people who, used hair dye or were hairstylists didn't have some sort of genetic predisposition or other exposures that could have also increased the risk for cancer? So it just becomes a really hard thing to study. And then, we also know that having previous exposure to chemotherapy or radiation can increase your risk for MDS. So we know that up to 15% of MDS is related to prior exposure of cytotoxic treatment. So we used to call this treatment-related MDS, and now we call it post-cytotoxic MDS, or post-cytotoxic therapy. And the cytotoxic is just this word that we use to describe any sort of treatment or any sort of therapy that can cause damage to our cells. We know that it can be caused by DNA damaging agents used to treat cancer. So a couple of DNA damaging agents are listed below, like alkylating agents or topoisomerase inhibitors or radiation. Now, it's important to mention that we do not discourage the use of these types of cancer treatments because they could potentially cause leukemias. We are mostly focused on actually wanting to treat the cancer. It's often really hard to get, worried about other cancer risks when, we want to treat the first cancer that is occurring, and then we can deal with some of those secondary risks, or we can be aware that those exist. Now, we also, of course, know that MDS is not the development is not within a vacuum. So there are multiple risk factors that often work together to cause MDS. And, I've gone through some of these, like smoking or benzene or, prior, chemotherapy or radiation, old age. And what I'd like to spend, the majority of this time talking about, and, what my true love in this world is the genetics of MDS. So we'll, of course, address most of that, but I wanted you to have a nice primer that it's often a combination of multiple events. And so these graphs here really are nice representations of this being a real multi-hit model. So, that it isn't often just a genetic risk, but it is the combined effort of a genetic and an acquired risk. So, I think that people love to talk about, is this nature versus nurture kind of thing. And even as a genetic counselor who believes a lot of different things are genetically acquired or genetically predisposed to developing, I still understand and recognize that this is not just a nature versus nurture, it's a combination of both. And so on the left here, if somebody is born with a lesser genetic risk and has an acquired risk, they may not develop MDS. But if they are born with a higher genetic risk on the right here, what you'll then see is there's less of an acquired risk that is necessary to then develop MDS. And the same could be true, if you have less of a genetic risk but more of an acquired risk, you could still develop MDS, but it's cumulative, it's all together. And so that is why it's often really hard to blame one thing or another. Now I'm going to go back to, this original slide that was discussing that it's caused by an accumulation of genetic mutations. So, that it takes one mutation to then develop a second mutation, and genetic mutations are what makes up all of our cancers. But what I will bring us back to is that, what if we are born one step closer to developing MDS? So what if instead of this genetic mutation being something that we acquired through our life, what if this is something that we were born with, that is part of our stem cells? And then, we're one step closer, maybe it doesn't take as long to develop this second mutation or develop this third mutation. And this is really the fundamental basis of genetic predisposition, and specifically genetic predisposition to MDS. So now that you've had kind of a nice biology lesson, I'm going to step into a history lesson. So, we know that MDS can be hereditary, but what is so surprising is just how recently we learned this. So leukemia was first described as familial in 1944. And some people might say, well, 1944 was a long time ago. And I don't disagree with that at all. But the first gene that was described to cause leukemia is a gene called RUNX1, even though it says CBFA2 here, it's now called RUNX1. This was not described until 2001. Now, I don't know about you guys, but to me, I'm like, oh, 2001, that was just a couple years ago, So it is impressive how short the history lesson is for MDS predisposition, because 20 years is really not a whole lot of knowledge. But what is amazing is that now when we're ordering genetic testing in 2023 for patients with MDS, I am routinely ordering genetic testing for over 100 genes. So even though the history is impressively short, we've learned a lot over the past 20 years. Now, We owe a lot of credit to Angelina Jolie in the space of hereditary breast cancer. And I think that pop culture and how we relate to pop culture and genetics is so relevant here, because the Angelina effect is a very well-known effect where patients are familiar with these genetic predispositions to breast cancer or ovarian cancer because of Angelina Jolie's widespread sharing of her story of having a genetic predisposition to breast cancer. Now, I will just mention that the knowledge of breast cancer genetics started in about 1994 when we discovered the first genes that were associated with breast and ovarian cancer, and that's BRCA1 and BRCA2. And we now know that about 5 to 10% of all breast cancer is hereditary, and 10 to 20% of all ovarian cancer is hereditary, and that almost all new diagnoses of these cancers do get some sort of genetic testing to see if there's an underlying predisposition. Now, I am still waiting for the day that there is a celebrity that is diagnosed with MDS and has some sort of genetic predisposition so that we can call it, the XX effect, maybe it's going to be the Hugh Jackman effect. I don't know. Preferably, I would love some sort of attractive male that we could then plaster on a bunch of posters. But at this point, I'll take anybody. So for the meantime, I will just continue on my, shouting from the rooftops that even though hereditary breast and ovarian cancer seems to be genetically tested more often and is more widespread in terms of offering, MDS is just as hereditary, and people should get genetic testing just as often. So frequently, MDS is due to an underlying hereditary risk. So I mentioned 5 to 10% of breast cancer is hereditary. Well, up to a third of MDS is hereditary. So why are we not doing genetic testing as often? And I think that the answer to that question is, there's lots of different reasons and variables, but, like I said, me and many other genetic counselors and providers are trying to increase awareness and trying to advocate for patients to get this type of testing, because even though we don't have a beautiful spokeswoman like Angelina Jolie, it still deserves its own opportunity for testing. So I wanted to share with everybody a recent study that was done, and this has really supported my advocacy to get this type of testing done, because essentially what researchers did was they sequenced, and by sequence, they did genetic testing of about 400 patients with MDS, as well as their related bone marrow transplant donors, so their siblings or maybe their parents or children. And what they did was they also examined patients across all subtypes of MDS and across all age groups. And this is very important because not only did they not restrict it to patients who are younger at diagnosis, which is an important red flag that we'll talk about, but they also did not restrict it to the different types of MDS or a single subtype of MDS. And so what we would call this is an unselected population of patients with MDS. And down below is basically just the methodology that they used in order to do this type of testing. And what they found was that 7% of patients had a predisposition identified. So 7%. So I mentioned that up to a third of MDS can be hereditary, and that's true in certain age groups, which I'll show you below. But what is also true is that 7% of unselected MDS has an underlying genetic predisposition. I'll remind you, 5 to 10% of breast cancer is hereditary. So it falls right within that realm. Now, what we found is that there are distinct risk categories based on the age of MDS diagnosis. So remember, MDS is typically a diagnosis that occurs at an older age because it takes a while to accumulate those genetic mutations. But we know that if you develop MDS in childhood, so under the age of 20, and I know that some people that are in their teens would hate that I said that this is childhood, but we'll just say childhood for ease during this presentation. But in childhood, since MDS is so uncommon, up to a third of MDS in childhood can be due to different genetic mutations. And then what we see in adulthood is anywhere from about 6 to 8% of MDS being hereditary. And we see different genetic mutations affecting adults diagnosed with MDS. And I'll talk a little bit more about this in a little bit. And then, kind of spectacularly, we see also in later adulthood. So even though you've had time to accumulate these genetic mutations in adulthood, we still see this being a hereditary disease in adulthood. And the star of this show in adulthood is really this DDX41 gene, which I'll talk more about in a minute. So really, this is just my primer, and this is just the slide, the picture that I love to show individuals and families and other researchers, and that, MDS is worthy of getting genetic testing as well. But I will just mention that, even though there is a high chance of identifying a predisposition, not every family has an identifiable risk. So, what this graph is showing, or what this pie chart is showing, is that there are many different genes that are associated with an increased risk of MDS. And these genes were discovered in this patient population, but there are still families that don't have a known genetic mutation. And I'll talk about what we do in those families. So this is just another graphical representation of MDS across your lifespan, with certain genes affecting, early life and in childhood, and then also in, young adulthood, and then in late adulthood as well. So, and these are, maybe genes that are associated with specific genetic syndromes. So we don't just see risks to develop blood cancers, but we see other things developing, or genes that are associated with mostly just risks to develop blood cancers or blood problems. So genes that are active only in the bone marrow. So to just start out by talking about MDS in childhood, as I've previously mentioned many times, this is a very rare diagnosis. And what we think is that all cases of patients who have pediatric MDS should undergo genetic testing. So this is more and more done in now 2023. But what I'm finding is that lots of patients that were diagnosed with MDS 20 years ago, 30 years ago, or even 10 years ago may not have gotten genetic testing. And just having a diagnosis of MDS in childhood is enough to get genetic testing. You don't need to have a strong family history of other cancers or any other features. You can just have MDS, and that is worthy of genetic testing. Now, the stars of this show are the SAMD9 and SAMD9L genes, and I'm not going to really spend a lot of time going through this slide. I know that this is a recorded webinar, so you're welcome to, review this and or look at some of the literature, but SAMD9 and SAMD9L have been these genes that have been newly described to cause MDS in early childhood. So anytime you see MDS under the age of 3, you should really be thinking SAMD9 and SAMD9L. Or if you see MDS and you see other features, like with SAMD9 gene, we see other things like infections or short stature growth restriction or problems with the adrenal glands, or maybe the patient has an XY karyotype, so meaning that they're born a male, but maybe their genitalia doesn't look like they're males. And so if you see other things beyond MDS, then you should be thinking, like, could there be a SAMD9 mutation? Or, with SAMD9L, what we see is that you can develop MDS and you can have neurological problems like ataxia. And ataxia is just kind of a funny way of walking. So it's a gait disturbance, or maybe we'll see problems with the cerebellum, the part of our brain where this controls our walking. And I included below here just some red flags. So when we start thinking about SAMD9, what would warrant genetic testing for SAMD9? And as I've mentioned, any pediatric MDS, especially those under the age of 3, but especially those with other genetic or other findings associated with it. And then we'll talk about GATA2 here, which GATA2 is also very similar to SAMD9, where we do see typically other things associated with it. And I think about GATA2 causing MDS usually above the age of 3, but below the age of, say, 40 or so, but it can occur older. And then what we'll see is, things like deafness or potentially warts, or interestingly, we can see these sort of opportunist infections. So infections that, people who have normal functioning immune systems would be able to fight off. But if you have a GATA2 predisposition, you can see, these infections causing significant problems, and you may even need to be hospitalized in order to prevent these infections, or to treat these infections, excuse me. And so, once again, with GATA2, we do see other things, but we kind of also just see MDS. And so getting genetic testing when you're diagnosed with MDS under the age of 50 is huge, especially in childhood, because of a gene like GATA2. Now, in early adulthood, MDS is still very rare, and I consider early adulthood anything less than the age of 50. So up to 20% of MDS in this age group or up to 20% of MDS in this age group can be hereditary, and any patient diagnosed with MDS under the age of 50 is eligible to undergo genetic testing. I'll just touch briefly on a couple of genetic conditions that can cause MDS under the age of 50, and one of them is Shwachman-Diamond syndrome. And this is what we call a bone marrow failure syndrome. So meaning that it can cause bone marrow failure, so the bone marrow to stop working, and it's a syndrome because it can cause other manifestations outside of the bone marrow. But what we're learning is that patients who have Shwachman-Diamond syndrome are at an increased risk to develop myeloid malignancies, and specifically MDS, as well as other findings like skeletal anomalies or problems with the pancreas. And importantly, this is inherited in an autosomal recessive manner. So meaning that parents are carriers, and when you're a carrier for a genetic condition, you don't typically have any sort of manifestations. But when you inherit both mutations in both copies of your SBDS gene, then that is when you develop Shwachman-Diamond syndrome. Lots of other genetic conditions that cause an increased risk for MDS are inherited in a dominant manner. And one of those conditions is a telomere biology disorder. I apologize that this got cut off up top here, but this just says disorder. And telomeres are these kind of funky things at the ends of our chromosomes that basically help our DNA to retain its structures through the process of replication here. And so it's just this repeated stretch of DNA that through cell division, and as we age, the telomere cap gets a little bit shorter, but eventually that telomere cap gets a little bit too short. And so our body recognizes that we can't do we shouldn't divide anymore, and we should undergo cell death. And some people, instead of having nice, long, healthy telomeres, those telomeres are much shorter, and these patients are predisposed to developing MDS. Telomere biology disorders are part of a syndrome, so we do see lots of other features of this disorder besides MDS. So up to 10%, or excuse me, up to 11% of patients with telomere biology disorders develop MDS, but a more common manifestation is pulmonary fibrosis, or interstitial lung diseases, or we can see cirrhosis of the liver, also called fibrosis of the liver, with or without alcohol use, or other things like premature hair graying. So importantly, telomere biology disorders are not diagnosed by just genetic testing, and we often use other things like telomere length measurement testing. So there are different tools at our disposal to diagnose genetic conditions. And then lastly, in this age group, but certainly not, this is not an exhaustive list of the genetic conditions that can cause young adults to develop MDS, is this RUNX1 gene. And this is an example of a gene that really only causes problems within the bone marrow. So we can see that when people are born with RUNX1 mutations, that this can lead to the development of MDS or other types of leukemias, like acute myeloid leukemia, which is the type of leukemia that MDS can develop into, or other types of lymphoid leukemias, like T-cell acute lymphocytic leukemias. And then on top of that, what we can see is that prior to the development of these blood cancers, we can see a long-standing history of low platelets, or maybe you don't have low platelets, but maybe you just have problems with how your platelets function. So maybe you bruise a lot easier than other people, and or maybe you bleed a lot easier, and that your body has a problem with clotting, or that that you bruise like a peach, so this is a common thing that I hear in my patients who are born with RUNX1 mutations. Now, lastly, and more importantly, because this represents a large portion of the patients that are diagnosed with MDS, are those that are diagnosed in late adulthood. And in contrast to other genetic conditions where, we get really concerned about breast cancer diagnosed in your 30s versus less concerned when breast cancer is diagnosed in your 80s, is that even though MDS is most commonly diagnosed in this late adulthood range, up to 9% of MDS in this age range can be due to an underlying genetic condition. But what is a little bit unclear is who should undergo genetic testing in this age range, because we do have a cutoff, which I'll explain a little bit in a minute. And the real star of this show is this gene called DDX41. So patients diagnosed with MDS at older ages, we see that up to 9% of these patients actually have a DDX41 mutation that they were born with, and that is what caused their MDS. And this can be a little bit hard to identify because not everybody who has a DDX41 mutation has a family history of DDX41 or has any other features that could lead us to think like, oh, this is going on right here. So I talked about, those other genetic conditions where we might see lung disease, or we might see, low platelets before developing the MDS. But with DDX41, we don't always see any of that. And then on top of that, because it's diagnosed at the age where we see sort of sporadic cancers developing, then, we don't always see that there is a red flag that we should be really worried about. So this is kind of the gene that throws us through the loop the most, because it can be the hardest to identify in some ways, and in other ways, the easiest to identify. And I'll explain what I mean by that. So who should undergo genetic testing? So when you're diagnosed with MDS, if you're diagnosed under the age of 50, you should undergo genetic testing. If you're diagnosed with MDS and have a previous history of another cancer, like breast cancer or prostate cancer, or maybe you have a family history of cancer, then you may be eligible to undergo genetic testing as well. Or what if you have MDS with those other symptoms that I've described, like pulmonary fibrosis, or having low blood counts, or having an opportunistic infection history? Then all of that warrants a genetic evaluation. So meeting with a genetic counselor, or discussing if genetic testing is beneficial for you. The question that I get is, why should you get genetic testing? So we think that, genetic testing, I try not to, have such tunnel vision, because I think that genetic testing can be beneficial for many reasons. And one of those is that, when you're diagnosed with a cancer, a lot of people wonder why they developed this. Was it something that I did throughout my life, or something that I didn't do throughout my life? Was it, I should have had more vegetables, I should have done X, Y, and Z. And so this may help you to understand why you developed MDS, and that, you were born with a larger genetic contribution to your disease. And so even though still practicing, some of those behaviors like eating healthy and getting exercise, you were more susceptible, and that is kind of what contributed. It may also help family members. So, some questions that come up are, what are my kids at risk for? What are my siblings at risk for? And so this can help to give you a little bit of a primer to understand what are they at risk for. And then on top of that, it may also help with some treatment decisions. So we know that when it comes to MDS, one of the treatment options is undergoing hematopoietic stem cell transplant or bone marrow transplant. And often when we're considering bone marrow transplants, we consider using a family member as a donor. Well, if this is something that's inherited, and something that your parents share, then your siblings could also share that. And so what we know is that donor-derived leukemias exist, where, somebody gets a stem cell transplant from a sibling. That sibling has a genetic mutation that predisposes them to developing MDS as well. We just went through all of that treatment, and, you went through that whole transplant just to get that same genetic mutation again and be at risk for developing a donor-derived leukemia. So many times when somebody is diagnosed with MDS, this is one of the main drivers that I'm doing genetic testing for, is to try to understand what is the best options for our donor situation. So that might be, instead of using a sibling, we might try to use an unrelated donor, or we might then test the sibling for the genetic predisposition that we identified, so that we know that the sibling is unaffected and is not going to give you that same risk for donor-derived leukemias. Now, what does genetic testing look like? And I like to really divide it into two categories. The first category is that if you have an active MDS in your bone marrow, or if you've had an allogeneic stem cell transplant, so meaning that you have undergone a stem cell transplant by getting a new set of bone marrow from somebody else, so that DNA is no longer yours, then the best way to do this genetic testing is with a skin biopsy. So this is where we're actually able to get genetic testing or a genetic sample from this part of our body where we're not expecting any sort of contamination from the leukemia. Because if we used a more traditional sample, like a saliva sample or a blood sample, we would essentially just be testing those leukemic cells. And that is really not what we're trying to do. So we're trying to get to this very important different DNA set, and that is why we would want to do a skin biopsy. But if you were previously treated for MDS and it's been, several years or a couple of years, and you're in remission and you've never had an allogeneic stem cell transplant, then you should be able to get genetic testing on a normal sample like a blood sample or a saliva sample. But your team may still decide that a skin biopsy is best, just to not worry about any opportunity for contamination. So what this looks like is that we're able to obtain a sample for genetic testing, and then we're able to perform sequencing where we actually look at the DNA, and then that is resulted as a genetic test result, which is a PDF result, often that's sent to the person who orders the testing. Insurance, that's a big question that comes up, is does insurance cover genetic testing? And I say it should, if you meet genetic testing criteria. So listed below are genetic testing criteria from NCCN guidelines. So this is the guidelines that many centers are using to diagnose and treat and treat blood cancers or other cancers. And there's a whole section in the MDS guidelines about who should undergo genetic testing for MDS and what testing should look like. So it's a really great resource for not only us that are in this field, because it gives credence to what we're doing, but also it helps provide justification for insurance to cover this type of testing. But many labs offer a cash pay price for genetic testing that's about $250. So genetic testing is no longer unattainable, and that's also why it's important for us to sort of spread this word that MDS is hereditary, is because it's no longer thousands and thousands of dollars to get the testing. Now, one of the questions that comes up is who should get genetic testing in the family? And so the analogy that we really like to use in genetics is you have this apple tree here, and this apple tree has lots of red apples, and there's one apple on the tree that is yellow. Well, in order to figure out why this apple is yellow, you wouldn't go ahead and test all of the red apples, right? You would test the yellow apple. And the same is true when you have MDS. So if we're doing genetic testing for MDS, we don't necessarily want to test the unaffected family members. We want to be able to test the person who developed MDS in order to figure out whether they have an underlying genetic predisposition. And then using that information, we may recommend other testing for family members, or if that testing comes back normal, then we wouldn't recommend anything above and beyond that. I wanted to clarify too that most patients with MDS do undergo genetic testing, but this is often what we call somatic testing. So this is testing of the tumor, and in this case, the tumor is the bone marrow. Looking for acquired genetic mutations that define the MDS or that help us understand what treatments might work best for you. And when we're doing genetic testing for hereditary risk, this is called germline testing. This is genetic testing that is, of those egg cells, the sperm cells, the DNA that we were born with that we can pass down. And so that is a very important distinction, because not everybody who has genetic testing that's performed on their MDS, this is not always considered specific for this other testing, this germline testing. So MDS testing that's performed on the bone marrow itself is often looking for chromosomal rearrangements or looking for genetic mutations. And this is a list here of some of the genes that we use at Huntsman for when we're doing genetic testing on patients with MDS. And I've highlighted and underlined these genes that we know are associated with an inherited risk. There are lots of genes on here that are listed in white that are not associated with a hereditary risk. So it is a big Venn diagram. It's great that genetic testing is done on MDS, but it's not always sufficient, and it does not replace at all a genetic referral or other genetic testing if somebody's diagnosed at a young age or has those other features or has a family history of other cancers. And then I included this table here, which is really just, when somebody has a GATA2 mutation, for example, we can see certain genetic mutations that are acquired in the bone marrow that can signify that there is some sort of predisposition going on here. And then we can also see other chromosomal abnormalities. And one of the ones that we see sort of recurrently is any sort of chromosomal abnormalities within chromosome 7. So if we see only one copy of chromosome 7, or if we see sort of rearrangements of chromosome 7, that can make us wonder, is there some sort of underlying genetic predisposition? But again, this is not a exhaustive list, and certainly every institution does different genetic testing. Now, just to kind of wrap up some of this information, MDS can be hereditary. I've explained that. I've gone through that, the situations in which MDS can be hereditary. But now there are these two other categories where there's sporadic MDS, which is most often what happens, where, these genetic mutations are accumulated through our lifetime, and it is not due to anything that we were necessarily born with. And then there's familial risk, which is when, maybe there are other people in the family who are diagnosed with MDS, but genetic testing has returned normal. So what do we do in those situations? So sporadic risk looks a lot like this. This is a pedigree here. We have one family member right here in the middle who developed MDS. But as you can see here, no other family members developed MDS. So of course, this could still be a situation where hereditary MDS affects this patient, but it's also could just be that this was one of the patients who develops MDS throughout their lifetime, and there's no underlying cause. Here's an example of a hereditary risk where there are multiple family members who've developed MDS, and in this particular family, let's say there's a RUNX1 mutation, and that's responsible for the MDS. And then here's an example of a family that has familial risk. So, it looks a lot like the family that I just showed you, but instead this family doesn't have a RUNX1 mutation or anything else. So the question here is, what are the risks for our family members if we do not have a genetic predisposition? And what I'll highlight is this study that was done, and this was done a couple of years ago on over 100,000 patients with blood cancers. And what they looked at was patients that have many different types of blood cancers. And the table here summarizes some of those, like, lymphomas or other chronic leukemias or myeloid malignancies. And this includes MDS. It also includes other malignancies. And then on top of that, there's a group of disorders called myeloproliferative neoplasms. So looking at some of these families, so what we're saying here is that everybody has about a less than 1% risk to develop MDS or to develop any other myeloid malignancies. So siblings of that patient or children of that patient have about double the risk in the general population to develop MDS. So that equals about 1.5%. Now, what if your family history is a little bit stronger? What if you have 2 first-degree relatives with MDS? So for this patient, he has a brother and he has a mother with MDS. So this patient's risk is about 4%. And, that's because of having these 2 first-degree relatives. And then instead, now this patient, the mom, her siblings still only have about 1.5% risk. Now, what are we doing for family members? So it's great to know what is my risk, but what are we doing? So do we screen for leukemia? That's a really hard question. Certainly when there's a genetic predisposition identified, we screen for leukemia. But what if there isn't a genetic predisposition? What are we doing? So you can always consider asking your primary care provider for a complete blood count. So it's always good when somebody has a baseline complete blood count. What if your low platelets have been persistent throughout time and you never knew about it? That's good for us to know. blood counts are not necessarily good as a one-time thing. They're often good in knowing, over time, what is your normal. So, getting a blood count once or twice or 3 times, just to see what your normal is. And then if you then develop some of these symptoms like weight loss or night sweats or weakness, or you're developing petechiae, those little bruises, then that's definitely a good time to seek, work up with a primary care provider. So symptom awareness and, looking at blood counts is really a phenomenal way to approach this. But there always can be more specific discussions, because this is not a one size fits all approach for everybody, especially in light of genetic testing. So I will wrap up by just saying that, in conclusion, genetic testing can be a beneficial way to understand why you developed MDS and who in your family may be at risk for MDS. There are guidelines for who should undergo genetic testing for MDS, which makes it easy to advocate for getting this type of thing. Now, genetic testing may help guide treatment decisions, like who is the best donor for your bone marrow transplant, or it may just be helpful for knowing some of this future information. And then lastly, genetic counselors are available to discuss this type of genetic testing. So this is what we do, this is what we love to do, and we're available as resources for you all. So I will leave you here with a picture of Utah this summer and my Twitter handle and my email address if anybody has any follow-up questions. So I hope that leaves enough time for any sort of questions that came up, and just really appreciate everybody's attention today on this important topic to me. So thank you.
Thank you so much, Jenny, for the wonderful presentation. And we do have some questions. The first question is, what is the best timing of genetic testing? When should patients be tested? And there was a separate question, and that question would be part 2 of that is, how often should patients be tested? And it's in reference to disease progression.
Yeah, good. Okay, I'll start by answering the first part of that question. When should you get genetic testing? Now, I think that this is really dependent upon the type of disease that you have and whether we think that there are going to be any sort of treatment decisions that would be changed if you have a genetic risk. So if you are going to transplant, then it is very good to get genetic testing as soon as you can in the diagnosis pathway. So I really like to see patients within the first 2 weeks of their diagnosis, just in case that influences any sort of treatment decisions, so that we have all of this information back by the time you do need a transplant. If you're not getting a transplant, or if you're going to have, more cycles of chemotherapy before transplant, then really any time through that process or after you're done with treatment is a good time to get genetic testing. Now, in regards to the second question, it's hard to know, exactly when during the time of progression do you get genetic testing, or how often you need it. But what I can say is that when it comes to genetic testing for an inherited risk or a germline risk, usually we say that if you've had genetic testing in 2023 and it was a comprehensive genetic test, meaning that we looked at many of the different genes and that can cause an hereditary risk, then you really don't need genetic testing again, this year or next year. You probably could get genetic testing again in 5 years. And that's because new genes are discovered or new technology is created, and that you may benefit from this updated genetic testing. But it's not something like a CBC where you need to get it, multiple times during a specific time period, because it's your DNA, and because it's inherited, it doesn't typically change. It only changes based on our new knowledge.
Great. Thank you very much for that. Our next question is, does genetic testing also let a patient know if they're at risk for an overlapping diagnosis, such as if you're an MDS patient, do you have the possibility of developing, say, a myeloproliferative neoplasm, an overlapping disease?
Good question. I typically don't think about these disorders causing an increased risk for you to develop like an overlapping MPN. Sometimes these genetic disorders may give us an insight as to whether you're more likely to progress, or how likely you will be to responding to treatment or responding to something like a transplant. Or sometimes this genetic testing could tell us how likely you are to develop another type of disease like pulmonary fibrosis. But I would say rarely do I use this testing to inform the likelihood of developing another type of bone marrow disease at the same time, like an MPN overlap.
Thank you. We have one more question, and this question is in regards to bone marrow donors. Do you know if during the donor testing process, do they undergo genetic testing to make sure they are not going to pass along anything to the recipient?
Oh, I love this question. This is really the million-dollar question, and a lot of people are looking at this question right now because standardly, no, there is not a significant amount of genetic testing done on donors. But there are current studies going on right now looking at this exact question, and we hope to have a better answer as to what this genetic screening process should look like for donors, what it should look like for really all types of donors. So, as somebody in the general population who wants to just be a bone marrow transplant donor, what should that look like if you're going to be an unrelated donor to somebody? And then on top of that, what should that look like if you're going to be a sibling donor? Should you also undergo genetic testing, and should you also get a family history done? And so there are enough cases of people developing donor-derived leukemias for us to really be super thoughtful about this. And so, it's a little bit of a Pandora's box. If you do a lot of genetic testing, you might find answers to questions that you didn't even know to ask. And that might not necessarily even be helpful information. But, you really want to find the sweet spot here for, getting the best information for the actual transplant, but then, not overdoing it. And so that's really what a lot of these studies are looking at right now, is trying to find that. So I hope that by the time we do another one of these presentations, I can answer that question with a little bit more certainty.
Thank you so much, Jennie, for sharing your time and your expertise with all of us today. This presentation was wonderful. So thank you so much for that. If you'd like to watch the session again at a later time, it will be available on the platform within the next hour. You can access this session by clicking on replay or by navigating the on-demand tab in the platform hub. If you have any additional questions, please send it to us in three ways. You can email us at help@aamds.org. You can give us a call at 800-747-2820, extension 2, or you can submit a question to our Facebook page. On behalf of the Aplastic Anemia and MDS International Foundation, I'd like to thank you for joining us today and making us your resource of choice for information on bone marrow failure diseases. Our medical advisory board and team are always here to support you and your family, as we've done for the past 40 years. This concludes our webinar.